Hello, everyone. Welcome to today's Congressional Neurosciences Caucus briefing. We really appreciate your attending today. I'm Matt Rizzo. I'm the chair of the Department of Neurological Sciences at the University of Nebraska Medical Center, and I'm also privileged to be the chair of the American Brain Coalition. Our event today is among a number of partners. Our ABC has come together today with the Cure Alzheimer's Fund and the Michael J. Fox Foundation for Parkinson's disease research. And we're partnering with the Congressional Neuroscience Caucus today on the critical need to better understand what causes neurodegenerative diseases and to develop novel and effective approaches for treatment and prevention. For your information, the Congressional Neurosciences Caucus was established in 2010, so it's a decade old. And the idea is to build awareness of the fundamental role that brain research plays in understanding ourselves and our society, to communicate progress and benefits of research, and importantly, to promote changes in federal policies that will help to advance this neurosciences research. We are hoping and expecting to be joined today by the Congressional Neuroscience Caucus co-chair. That's Earl Blumenauer. Not sure he's with us as yet, but he co-directs the caucus with Congressman Kathy McMorris Rogers. He represents the 3rd Congressional District in Oregon, and that includes Portland, and I think also part of the Mount Hood National Forest. So he's got people and maybe even a few bears. He's a lifelong resident of Portland. He was first elected to the U.S. House of Representatives in 1996. He used his time in Congress and is a member of the House Ways and Means Committee, working very effectively and for a long time to promote healthcare access and also environmental reform. He's also been a champion of patients suffering from neurologic and psychiatric disorders, and with what we're going to talk about today, developing understanding of the basic mechanisms and combating cures and developing cures to combat these disorders. So we're deeply grateful for his leadership, and we hope to hear from him if he's able to come on. Congressman Blumenauer, if you're on now, please come on. But if you're not, what we will do is go on to our educational part of this effort today. And I would start with Dr. Shane Littlelow. I want to say that neurodegenerative diseases from Alzheimer's to Parkinson's and related disorders affect millions of people worldwide. The risk of disease increases with age, and of course, people are living longer than ever. And so that means many, many millions more of people in the US and worldwide are going to suffer from Alzheimer's and Parkinson's disease and related neurodegenerative disorders. So I want to say again, we have 2 great speakers today, Dr. Penelope Hogarth and Shane Littlelow, and Shane's going to kick it off. He's an assistant professor in the Neuroscience Institute and Department of Neuroscience and Physiology at the NYU Langone Medical Center. His work focuses on understanding the interactions between glial cells and neuronal degeneration and dysfunction. So his laboratory is doing cutting-edge research on what molecules moderate these critical interactions, on the role that the astrocytes play, these connecting cells, in initiating and causing progression of disease and Alzheimer's disease. He's pioneering these models in a number of ways, both in the laboratory, in vitro, and in vivo models. And the goal is that they replicate what's actually going on in normal processes of human aging, but also in disease. So we're thrilled to have you here today, Dr. Littlelow, and please take it away.
Thank you very, very, very much for that kind introduction, Matthew. And I also, I thank all the members who have come online to interact with us today and to listen and learn a little bit about the science And I particularly like to thank Representative Blumenauer and also my collaborator in crime here, Penelope, who's also going to be telling us about some amazing work. So as Matthew mentioned, I'm based in New York City at the NYU Langone Hospital, where we have a large research aim as well. And my lab is really interested in understanding neurodegenerative disease. And we're sort of disease agnostic. We work in lots of different spaces. The vast majority of the work we're doing at the moment is in Alzheimer's disease. But we're really trying to understand the nuts and bolts of the disease from a different angle. And so I want to take you on a little journey today and just tell you a few vignettes about some of the difficulties with investigating this and some of the exciting discoveries that the amazing scientists in my lab have discovered over the years. So I always like to start with just very broadly just to discuss exactly what is neurodegeneration. I think, you know, often scientists and clinicians, we forget that we bandy around these terms that have a lot of information behind them without describing them properly. So that we're all on the same page, neurodegeneration or neurodegenerative diseases are really an umbrella term, a catch-all for a whole range of different conditions, which primarily affect neurons in the brain. But we also talk about neurodegenerative disease in the context of death of other cells, such as death of oligodendrocytes, which are another type of glial cell in diseases like multiple sclerosis. And neurons, as we will get to, are obviously the building blocks of the brain and of the spinal cord. And collectively, we call those two masses of neural tissue the central nervous system. So I will interchangeably use central nervous system or CNS or brain throughout the talk, but know that I'm meaning the same thing, more or less. And generally, neurons don't reproduce or replace themselves. We go through a period of development in utero and after we're born. And although there is small instances of occurring, it is largely that once the neurons are damaged or dysfunctional or die, it's very difficult for us to regenerate them without intervention. And this is neurodegeneration. This is a perfect image of what happens in the brains of human patients. So on the left-hand side, I have here just the brain from a human patient that we would call non-symptomatic or otherwise healthy and normal on the top left. And on the bottom left, we just have an image of a slice that's been taken through that brain so that we can see all the structures that comprise the human brain. And on the right-hand side here, we have a brain from a patient who unfortunately passed away due to Alzheimer's disease. And I'll just put a call out to thank all of those donors who have enabled us to do a lot of this work across many, many labs throughout the US and the world who have donated their brains and other tissues. But we can see that the brain here is obviously looks very different. And when we cut this brain open, we can see that there are big holes in that brain. There's lots of pieces of the brain missing. And that is degeneration. That is the death or the loss of neurons. And this is what we're going to be talking about today. So just a few very quick facts and figures, and I'll rush through these. But you know, we can talk about lots of different diseases, and Penelope will talk about this as well. But you know, in the case of Parkinson's disease, there's around 60,000 or so new diagnoses in the US alone, and more than 10 million patients around the world. If we think about multiple sclerosis or glaucoma, the numbers jump up a little bit, but there's around 1 million cases or more in the US alone for multiple sclerosis, and around 3 million cases in the US for glaucoma, which although is a disease of blindness, it does have to do with the degeneration of neurons that are actually based in our retina. And then what I'll spend a lot of time talking about today is Alzheimer's disease. I'm sure we all have heard of and know someone or a friend or a family member who has unfortunately passed because of Alzheimer's disease. But there are around 5 million cases or patients in the US alone. And it's predicted that around a third of seniors, you know, these are people over the age of 65, are dying because of Alzheimer's or other related dementias. So this is a huge number of patients, and it's only going to grow going forwards. And from a cost perspective, which is really important, especially from a government financial side of things, you know, just this year alone, in Alzheimer's alone, the cost to the public is around 300 billion, and that's expected to rise in the next 30 years by about threefold to over $1 trillion. And although it's very easy to understand the money, although I've never seen that much money in one place, there's also a sort of a hidden cost to this. And the hidden cost comes from unpaid carers who are involved in taking care of friends and family members. And it's estimated there's around 16 million of these unpaid carers in the US alone as well. So this really does impact our society, and that's only picking one of these diseases, and there are numerous neurodegenerative diseases. And the other thing I want to say is, you know, it can get really depressing and it can get really tough just sitting at home and reading the news headlines and seeing it online or seeing it in your favorite news channel, and everything seems to be doom and gloom. And I've picked out a few news headlines here just focusing on Alzheimer's disease, and it can at times feel like this is an insurmountable problem, that it's too much, it's too complicated, and we're not really making any headway. And I'm here to tell you that it's not all doom and gloom. It's not all darkness. A few things have changed. And the main thing that's changed is, at least for me, I'm getting a lot more of my news from Twitter because it's just very rapid and very quick. And from a scientific standpoint, a lot of scientists use Twitter to actually get information out to the public. So I highly recommend that if there are diseases or researchers that you're interested in, check them out on Twitter. And the other thing that's important to keep in mind is, just because things fail doesn't mean, or just because drug trials fail, doesn't mean we haven't learned anything about the disease. Every single clinical trial, every single drug that is made teaches us something about the disease that drug was made for, or that clinical trial that didn't reach muster. So we have learned a whole host of things about Alzheimer's disease from all of these clinical trials. Now, what have we learned, and what is my role here as a scientist? Well, my role is to investigate science, but it's also my role to disseminate that science to other scientists and to the general public.
Shane,
and so one of the things...
May I interrupt you just for 1 second?
Please.
The congressman has been able to shake free and join us. Can we pause just briefly?
Perfectly, I can.
And I just want to say, you know, we're grateful the congressman is here, and if it's good, it's worth waiting for. So I just want to give Congressman Blumenauer, who's been a great friend of ours, a time to say a few words. As I mentioned before, he's a long-standing member of the House of Representatives since 1996. Again, advancing healthcare and environmental reform for a very long time. A tireless champion for combating neurological and psychiatric disorders. And I mentioned before, Congressman, that you're representing the 3rd district that includes Portland and also the Mount Hood National Forest, leading to speculation that you also had some bears among your constituents. Maybe that's not true, but anyway, we're delighted you're here. And if you would say a few words, we'd be honored.
Well, thank you. And I apologize, Shane, for interrupting the flow. I appreciated what you were saying and putting in perspective what the stakes are for our neuroscience research. Just what you talked about in terms of Parkinson's or Alzheimer's, the costs literally are incalculable. And the benefits for our being able to move forward with the vast array of neuroscience research are probably the biggest payoff for the individual dollar that we invest. We're in the midst of the COVID-19. It is a crisis that we're ill prepared in part because we've got an administration that is just appears not, well, I won't get into the politics. It's changing. In 1,000 hours, we'll have a fresh start with the new administration. But neuroscience and the need for this research is something that shouldn't be at all partisan and really shouldn't be that questionable. We all know people in our lives that are struggling with neuroscience-based conditions. Addiction, you mentioned MS, that was 60,000.
I think you're muted, Congressman.
I got so excited, I jumped in. Let me say I did apologize to you, Shane, for interrupting the flow of your fascinating presentation and putting in context what we're talking about here. All of us know people who suffer from neurodegenerative diseases. This is the area that probably affects more individuals than any other. The costs are incalculable. The payoff, if we're able to make even modest progress, are tremendous. I mean, you referenced the 5 million people suffering from Alzheimer's. If we're able to delay the outset of Alzheimer's for 4 or 5 years, that will have a tremendous impact, since so many of these folks will probably pass away in that period of time, freeing up resources, avoiding the pain and dislocation on family members. There's probably no greater area for our to direct federal dollars than neuroscience research. I appreciate your working with us, staying the course, being able to work with the Neuroscience Caucus to spread information about how important this is. I find when I engage with colleagues and we get their undivided attention for 10 or 15 minutes, which is really hard to do back here, trust me, that it makes sense to them. And we've had pretty good success, even in an era of budget restrictions. I'm hopeful that people will realize, as we're trying to recover from COVID-19 and the economic distress and the upset, that continued investment in neuroscience research actually helps the economy, helps advance a healthcare agenda, and is something that brings people together. I'm sorry for the delay. I was on the floor voting, and I'm going to sprint back over in a minute. But I did want to be able to listen a little bit to what you're doing, look forward to following your progress, and look forward to working with you all as we begin a new session of Congress, where we have an opportunity with some new people to be able to encourage them. And most important, I will say, that the Neuroscience Caucus concentrates on the super smart young people who actually run Capitol Hill. These people are our major target. Being able to inform them with the caucus activities, with briefings like this, they're the secret weapon, and they will be the key to success. So I appreciate you all participating in this process. I look forward to starting a new Congress with some expanded participation in the Neuroscience Caucus, and welcome the information you're giving us. Thank you so very much.
Thank you so much, Congressman Blumenauer. We know that you're really busy, and there's a lot of work to do to sort of set the ship on the right course and help all of the patients that we serve. Thanks so much. And Shane, you're up. Sorry to interrupt. Take it away.
Absolutely no problems at all. I don't think I've ever been interrupted by such a wonderfully eloquent speaker before. So I really appreciate that the Congressman is here, and I would just like to reiterate, I think, you know, the bipartisan nature of these problems across the US and the world, and the cost is something that we keep in the back of our minds all the time when we're working on this. And it's also one of the things that we spend a lot of time doing in our outreach endeavors as well. As, you know, as I was saying earlier, we're really trying to make sure that we make the information that we learn more accessible to the general public. Your taxation dollars are funding a lot of the research that we do, so we're incredibly indebted to you as well. You are a part of this endeavor. And some of the questions I always like to ask people on the street, students who may come from high schools to learn about regeneration, or now I'll ask the Congressman as we go through, is, you know, what do we know about the brain, or what do you know about the brain? And generally people say, well, it's in our head and it makes us live and love and feel and walk and do all those things. And then I ask people, well, what do you know about neurons? And quite often people say, oh, well, they make up the brain and there's something about electricity and, they make things work. And then the next question I ask everyone is, what do you know about glia? And I would say nearly 100% of the time there are crickets and no one knows what's going on. And then I have to ask myself, should these people even care? Should you even care about what I do in my lab working on these strange cells in the brain? And I would say you should care because although we know a lot about neurons, they make up less than half of the cells in the brain. The rest of the cells in the brain are made up of these supporting cells, these glia. And just in the last few years, we had a couple of really big breakthroughs investigating these glia in the context of neurodegenerative diseases. So with collaborators at Johns Hopkins, we worked out an interaction between the immune cells and glia that drive the death of neurons in Parkinson's disease. And there's now a clinical trial ongoing as one of the first therapies targeting this mechanism in human patients, which is great. Along with collaborators at the OHSU School of Medicine in St. Louis, we discovered that one of the highest risk alleles for Alzheimer's disease, ApoE, is actually driving the dysfunction of these glial cells, in particular astrocytes, leading to neurodegeneration in that disease. And very early on, when I first moved to New York, we had discovered that there is a form of these glia called astrocytes that secrete a toxic molecule that can kill neurons. And it's actually the interactions between the immune system and peripheral infection that may actually drive this, these early stages of the disease and early stages of degeneration. So I guess the question is, what is an astrocyte, right? And what is a glial cell? So we're going to focus all on astrocytes for the remainder of the talk here. Astrocytes are star-shaped cells in the brain. Astro coming from the Greek for star, cyte coming from the Greek for cell. And so I have a cartoon here in green of an astrocyte with lots of these processes radiating out for it. And astrocytes are really important in the development and the normal functioning of the brain. They essentially are the chefs and waitstaff and the waste collectors for neurons. So they provide food, which I would call trophic and metabolic support, but essentially they're providing nutrients and food to neurons. They provide the ability for these neurons to connect to one another so that signals can move throughout our brain. We call these synapses, but they're just little connections like holding hands. They interact with the immune cells, and they also interact with the blood vessels so they can shuttle waste out of the brain and take more nutrients in that we're eating and consuming on a daily basis. And this is just a small number of the functions that these astrocytes have that make our brains work and ultimately make you, and they're the one cell that you cannot remove from the brain. If you remove them, unfortunately, you would pass. But in the context of disease and injury and infection, either bacterial or virus, as the congressman brought up, these astrocytes change and they become what we would call reactive astrocytes. And they can do a number of different things. So one option they have is that they can become really ultra supportive and they can support the health of these neurons. They can stop the infection from coming in further. But what we'll talk about in the context of neurodegenerative disease today is maybe these astrocytes can just become a little less competent or a little bit less good at their job. And so maybe they don't really provide enough food, or maybe they don't take away enough waste. And these neurons here in brown just become a little bit sick, meaning that they don't function as well as they should. Or there's the possibility that these astrocytes can release a molecule that actively kills these neurons. And this is something that my lab discovered a number of years ago, and we've been investigating really heavily since then. So what do astrocytes really look like? They're not cartoons in your brain, obviously. They look a little bit more like this. These are some astrocytes that we're growing in a dish in the lab. And you can see, although they're a little bit more complex, they still have this very star-like morphology with a center here with lots of processes radiating out. And they can transmit signals to one another similar to neurons, but they're a little bit slow. These are quite healthy neurons. We can also look at healthy neurons here, healthy astrocytes shown here in green. But when they become reactive and they start secreting this toxin, I've just labeled them here in red for you. So they change the way that they look and they change the way that they interact with the neurons and other cells in the brain. But of course, this is in a two-dimensional space. We like to think about the brain as a three-dimensional organ. So as Matthew pointed out earlier, this looks a little bit more like a Christmas tree, but this is actually just a three-dimensional reconstruction of these astrocytes in your brain. And it's just going to do a little bit of a fly through here. And we can see rather than being these flat cells, they're actually very bushy, like a sea sponge you might find on the beach or have in your bathroom. And in the human brain, there are around 100 billion neurons. That means there's about 1,000 trillion connections between each of these neurons. And each astrocyte is interacting with around 1 to 2 million of these connections or these synapses. So they're really modulating how the neurons are communicating to one another and making sure that they have everything they need to function normally. So it's a very, very complex system, the brain. And this is why I think it's been really complicated for people to understand exactly what's going on in the context of these diseases, just because of the enormity of these cells and the enormity of the interactions between them. So let's talk a little bit more about some science, and I apologize if this gets a little bit in depth, but please at any time you are more than welcome to drop a question in the chat, or I'm going to leave my contact details in the top right hand side corner of these slides. You can reach out to me personally if you'd like. But I'm going to stick to Alzheimer's disease for a little. Quite often what will happen when we're studying these diseases is we'll do what's called a genome-wide association study, and that is we sequence the genome of lots and lots and lots of people, and we look for mutations in genes that may be associated with those diseases. And what comes out of it is a boring plot like this, which we call a Manhattan plot, because it looks a little bit like the city skyline. This would be the financial district, a little bit of Midtown. This is Millionaires or Billionaires Row here, just south of the park. And you can see that some of these genes that may be familiar to you, like ApoE or clusterin, that are associated with these diseases. And so what my lab likes to do is say, well, if these genes are associated with disease, what cell expresses those genes? And can we understand the change in the function of those cells? And remarkably, the vast majority of these genes are actually not expressed by neurons. And this is just what we would call a heat map. The red color is showing that there's a lot of the expression of that gene in that cell type. The blue shows that there's not very much. And maybe 20% of these genes are expressed in neurons, while 3/4 of them are expressed in other cell types, the glial cell, the astrocytes, which we'll talk about today, the oligodendrocytes, which are the cells that die in multiple sclerosis, and the microglia, which are immune cells in the brain. It's the interaction between the microglia and the astrocytes that we're really interested in. So what my lab actually does is say, well, we're interested in what the physiological role of these astrocytes are, and we're also interested in how their functions, how their interactions change in the context of all of these different diseases. We're also really interested in how heterogeneous this is, and by that I mean how different this response is across different patients of different sexes, in different regions of the brain, or in different diseases. And we're also really interested in when this actually occurs during the disease. Does it occur very, very early before people even know they have the disease? And could we use this really importantly as a diagnostic tool or as a way of treating people pre-symptomatically to make sure that we just don't have these patients? Or is it something that occurs very, very late in the disease and we might be able to just only use it as an ability to stop the later stages of progression? And something we won't talk about today, but we're also really interested in why this happens, why evolutionarily we have this response. Is there actually some beneficial goal here with these cells responding this way? So a number of years ago, we were really interested in this, and just to sort of dig into the science some more, we were interested in how an astrocyte which interacts with all these cells may become reactive. And so we discovered that there's many, many, many different insults that can respond or cause a response in these immune cells. And in the brain, those immune cells are called microglia. So we're in the midst of a pandemic now. It's really interesting for us to discover how COVID-19 may be mounting a response in the CNS. We could also be talking bacterial infections, or we could be talking about the response of pathogenic proteins like amyloid or tau and how they respond. But these microglia then release a whole host of molecules that activate these astrocytes, and then these astrocytes start doing bad things. They're kind of like the hitmen for the microglia mafia. So some of the things they do is they start killing neurons, which we talked about. They also start killing the connections between these neurons, getting rid of these synapses so that even if the neuron is healthy, it can't communicate with the brain properly. We also know they kill oligodendrocytes, which is the cell that dies in diseases like multiple sclerosis. We know that they have implications of interacting with the blood vessels. They also feed back to the microglia, so you get this vicious loop happening where an angry microglia makes an angry astrocyte, which makes an angry microglia, which makes an angry astrocyte, and the cycle keeps going. And they can also feed back on themselves or their neighbors and cause dysfunction across these cell types. So do we even know? We do a lot of this work in animals, but do we even know if they're present in human patients? Well, we do. We can actually go in and what we're looking for here is these tiny little red dots, which is a single gene that is only expressed by these neurotoxic astrocytes. And we know, once again, through great donations from patients and their families that have passed away, we can look in the brains of patients in post-mortem, and we can see that in patients with Alzheimer's disease, there's a larger number of these cells than there are in non-symptomatic or control patients. But we also know this is true in Huntington's disease and amyotrophic lateral sclerosis or Lou Gehrig's disease, in Parkinson's disease, in multiple sclerosis, and in normal aging and a whole host of other diseases as well. This is a very, very common response that is happens across all of these diseases. So the question is, how do I see neurodegeneration? So quite often in my lab, I see neurodegeneration in a very different way. I see neurodegeneration looking down a microscope at cells in a dish. And what we have here is some cells, some neurons. And on the left-hand side, we have neurons that are healthy, that are growing with healthy astrocytes, and they're happily growing along there nicely. But on the right-hand side, we have healthy neurons that are grown with angry neurotoxic astrocytes. And you can see very rapidly, these astrocytes are able to kill these neurons, and they start to burst and blow up, and all of their beautiful processes start retracting. This here is the crux of neurodegeneration at the cellular scale. And we have the ability now to stop these neurons from dying, to stop these astrocytes from becoming reactive, and to stop these astrocytes from releasing these toxins. And we hope this will be a really exciting endeavor to make new therapies for patients with these diseases. We can also study this in mouse models. And so this is actually a mouse model of Lou Gehrig's disease, or ALS. And what we're looking at here is the ability of these mice to stay alive, so the survival. And in this particular mouse model shown in red, these mice normally die at around 3 or 4 months. But this blue curve here shows us that if we stop the formation of these neurotoxic astrocytes, we can keep these animals alive for about 50% longer. And the Congressman talked before the importance of even an extension of 4 or 5 years of life in a human patient, which may represent only 5 or 10% of their life. Here we've got the ability to increase the lifespan in this particular model by 50%, which is a huge increase. And of course, we're really interested, is this just happening in this animal model? And does this mutation have any effect on the ability of these astrocytes to kill in human patients? And so we can jump back and forth and look at this. So here we have just some staining in human tissue showing that in normal healthy patients, they don't have these neurotoxic cells shown here in the C3 marker. But we can see in patients with a range of different mutations, we can see there's a number of these red cells, which are these killer cells, and they're located in regions where these neurons would die, in the spinal cord and in 2 brain regions, which we call the medulla and the motor cortex. So skipping forward here, I'm just going to quickly say that we think about diseases having this trajectory, and in this case, we're looking at Alzheimer's disease, where we tend towards dementia, where we have a region where we have an interventional window, where we can detect memory dysfunctions, death of neurons, and also some pathological proteins like amyloid and tau. But we see that this glial immune dysfunction happens very, very early. And so while at the moment we only have this small window to interact, we actually are hoping that we can provide a much larger windows that we can treat these patients, that we can detect earlier, and we can extend this as further and further back as possible. So I'm going to stop here. So we have time for Penelope to speak, and so we have time to get through questions as well. But ultimately what we're doing is we're looking at understanding this disease from the context of a different cell type that is changing very early and driving the death of these neurons.
Thank you so much, Dr. Little, for your creative and deep exploration of the role of astrocytes, something people don't even know about or think of in the generation of neurodegenerative impairments. We'll move on now to Dr. Penelope Hogarth. We are so pleased that you're with us. Dr. Hogarth joined OHSU, that's the Oregon Health and Science University, the Movement Disorders faculty in 2000. She's a professor of neurology and medical genetics there, and she's also a clinician scientist at the VA. She's an expert in all sorts of movement disorders. Her main research focuses on Huntington's and Parkinson's disease. She leads a deep brain stimulation, that's DBS cooperative study, and that's comparing best medical management versus electrical stimulation of different parts of the brain called the subthalamic nucleus and the globus pallidus for Parkinson's treatment. And she's also interested in very novel treatments of Huntington's disease as a member of the Huntington Study Group. So Dr. Hogarth, I hope I've got that all correct. Assuming so, you're on. Thank you.
Okay, so the first challenge is to see whether I can actually share my screen. So let's keep our fingers crossed and try this. Earlier when we tried it, we had a little bit of a challenge. So Katie, you're going to let me know if the view is correct when I go to full screen.
Not exactly that view, no.
How about now?
Not exactly.
Okay.
I think if you swap displays, you might be okay. Try that.
I think that's going to put my
There we go.
There.
Perfect.
You're good.
now you have full screen.
Yes.
Yes.
Okay.
Thank you.
So my apologies to everybody for putting us through the technological challenges that are common in these times. It worked perfectly when we tried it first time this morning. And then of course, right before we went live, things got a little messed up, but we're here now. So thank you for the introduction, and thank you to the American Brain Coalition and the Michael J. Fox Foundation for inviting me to be a part of this. And of course, thank you to the Congressman. I think he's probably left us to go cast important votes, but just to let you know, he's actually my representative in my very district. So, and he's a wonderful supporter of our university's neuroscience program. He's been a very vocal and wonderful advocate for neuroscience research. So what I'd like to do this morning is I'm not going to talk so much about my primary research, which has actually shifted a little bit from Huntington's disease to a group of rare neurodegenerative diseases that affect children and adults. I'm in the genetics department now. But I'm going to talk today about Parkinson's disease and give some perspective on where we are and where we're going, as I see it. So let me see if I can. So what causes Parkinson's disease? I'm going to fix one other thing here, just so I can see everything that I need to see. So what causes Parkinson's disease? At its simplest mechanistic level, Parkinson's is the loss of the neurotransmitter dopamine. So when you look at this area under the microscope, this area that's shown in the black box there, you can see these abnormal accumulations of a protein called alpha-synuclein. And if you look carefully, you can see the same protein in other nerve cells in the body, for instance, in the gut, which gives us some insights into kind of where the disease might start. So pathologically, if you look at this part of the brain on the black box, and can you see my cursor in the screen share, if I'm moving it around or not? You can. Thank you for the nod, Jane. So when you look at this area under the microscope, in a normal person, you actually see this sort of dark area that is was originally called by the pathologists in early days the black substance or substantia nigra. And what has been noticed very early when people started looking at the brains of people who had died with Parkinson's disease was that this black substance had sort of disappeared or faded. And that gave us a place to start a couple hundred years ago of how to understand the disease. So it is this area where the dopamine producing cells live, and they die off in Parkinson's disease, leading to the loss of this neurotransmitter dopamine. So pathologically, you see these clumps of this abnormal protein called alpha-synuclein, and this has given us a target for developing therapeutics, which I'll come to in a little while. So what does it look like clinically? Parkinson's disease causes these motor symptoms that many of you are probably familiar with, this shakiness, a tremor, slowness, a type of stiffness called rigidity, and also really one of the most disabling symptoms is a loss of postural responses, meaning that the person has trouble with gait and balance. And then in addition to motor symptoms, there are these cognitive changes that can lead to some problems with memory, but in particular with executive function, meaning that person has trouble perhaps with over time with multitasking or managing to plan things well. And then there are autonomic symptoms that can evolve in addition to the motor symptoms. They might have trouble with blood pressure regulation. They might have trouble with constipation. These are very common things even quite early in Parkinson's disease. A significant number of people with Parkinson's later in disease will develop quite significant cognitive changes, really frank dementia, and sometimes neuropsychiatric disorders. So in advanced disease, people can experience psychosis, but in early disease, dopamine is a chemical that is not only responsible for fluid coordinated movements, but it's also responsible for motivation and feelings of pleasure, joy, if you like. And so it's very common early in disease even for people to say that they feel sort of flat. They may develop mood disorders. So it's really quite a disabling disorder. And interestingly, I mention this now because I want to come back to it when we talk about biomarkers and how we're kind of exploring Parkinson's treatments for the future. Quite a lot of people with Parkinson's report a lot of loss of sense of smell, and some report sleep disorders, a particular sleep disorder called REM sleep behavior disorder, where the person would act out their dreams instead of being still during dream sleep. This is very hazardous, not only to the person, but to their bed partner. They often might injure themselves or the bed partner. And those symptoms, loss of sense of smell and REM sleep behavior disorder, can occur many, many years before the onset of movement problems. And this is something that I want to come back to because it gives us some targets to look for before the disease starts. So Parkinson's, there's about 1 million people living in the United States with Parkinson's disease, and about 7 million worldwide. Dr. Littleow's disease, Alzheimer, definitely wins the day in terms of the most common neurodegenerative disease, but Parkinson's is actually the second most common. It's the most prevalent disease. So it has an overall prevalence of about 17 per 100,000 in the US, but this really changes very dramatically with age as one ages. So in the fourth decade, it's just about 41 per 100,000 people. But after age of 60, the prevalence rises very dramatically and reaches about 1900 per 100,000 after the age of 80. So really quite a common disease. Many of you probably know at least one person with Parkinson's in your circle, I would imagine. So what this translates to is then, as the population ages, the prevalence and incidence is expected to increase dramatically for Parkinson's disease. And this is also true, as I think Dr. Littler pointed out, the burden of Alzheimer's is going to increase over time as the population ages. Interestingly, Parkinson's is about 1.5 times more common in men than in women. We don't fully understand all the reasons for this, but it's an active area of research. And then just to talk a little bit about the economic burden of Parkinson's, Parkinson's is produces a very substantial economic burden to society, to payers, to patients, and to caregivers. A recent study found that the total economic burden of Parkinson's disease in 2017 was around $52 billion. And this amount was split roughly evenly between direct medical costs and indirect costs. And with the US and indirect costs being things like lost wages, lost wages of care partners, for instance, disability payments, those kinds of things. So really very substantial costs to society, to families, and to payers. With the US prevalence of the disease projected to rise to about 1.6 million by 2037, these costs will rise to a truly staggering $79 billion. That is what the projections are. And I just point out, since we're talking to a congressional caucus, that the Medicare program bears the largest share of these excess medical costs because most patients with Parkinson's are over the age of 65. And these kinds of realities are true for Alzheimer's disease too, which we've been hearing about today. Hold on just 1 second. I want to move my view here. All right. Sorry, I'm having trouble getting my cursor back to where I need it. There we go. So what's the current landscape in Parkinson's disease? So drug development to treat Parkinson's has largely focused on symptom management to date. That is changing very rapidly. But there's still a significant focus, of course, on managing the symptoms of Parkinson's disease because these are profoundly disabling. Really one of the amazing, most incredible medical drug inventions ever made was the development of levodopa, which is a synthetic form of dopamine to treat Parkinson's disease, to replace the dopamine neurotransmitter that is lost. And for those of us that have seen people with advanced Parkinson's disease who are in the so-called off state, where their medications have worn off completely, and they really have very little dopamine floating around in their system, to see somebody who is dopamine responsive take their medication and switch back on. We use that terminology because that is sometimes just what it's like. It is really like flipping a switch for some people. They can suddenly move again. This was captured, I think, in a movie that's now becoming rather dated, I think, but the Awakenings movie with Robin Williams. If you haven't seen it, it's an oldie but goodie, and it is well worth seeing it because I think it actually really does represent kind of what those early days were like with levodopa coming available and having the ability to truly unlock these locked up patients. So symptom management focuses not only on the movement problems, but that is where most of the drug development stands. So there are medical approaches that focus on the direct replacement of the lost dopamine. This is levodopa and related compounds. There are drugs called dopamine agonists that are kind of like a key that fits the same lock as dopamine, and they can have the same benefits for movement challenges. I would say that the research in this area is really, there's been some very novel approaches to alternate methods of delivery, because one of the curious things about the biology of our natural native endogenous dopamine is that it is delivered in a continuous fashion, whereas what we do when somebody takes a drug is we deliver that in a pulsatile fashion. And it turns out that the brain does less and less well with that over time, the fewer native dopamine producing cells it has. And so unless we can deliver dopamine in a continuous way, exogenous, external dopamine in a continuous level way, what we start to see as the disease advances is what we call motor fluctuations. And this is where the person goes from being rigid and stiff and unable to move to being on, so the drug kicks in, but then they kind of go too far and they start moving with involuntary movements that we call dyskinesias. And some of you may have seen this with various public figures who have advanced Parkinson's, may have seen examples of dyskinesias. These can be really very disabling. So the focus into symptomatic management of Parkinson's, I think some of the coolest stuff is around different delivery systems that try to mimic the native dopamine production. There's sublingual forms. There are these pumps now that try to deliver a continuous dopamine. That's one of the areas of research, continuous dopamine delivery. There are novel delivery systems that try to get past the challenges with the part of the gut where dopamine is absorbed. Because the gut is affected in Parkinson's, it slows down like everything else. When you take the drugs by mouth, they don't always get absorbed in the part of the gut that they're supposed to be. So there are now some novel delivery systems that deliver the drugs directly to that part of the gut. There are also, for people who switch off very brittly, there are some inhalation systems. There's a sublingual preparation now. And there are patches, skin patches, that try to mimic the native system by a continuous delivery. There are non-medical strategies to symptom management. These include the traditional things like physical, speech, and occupational therapy. But I would say one of the coolest things that has finally gotten prime time attention is the value of exercise. There is absolutely no question that exercise improves the symptoms of Parkinson's disease, and there's quite a lot of emerging evidence that it may actually slow the progression. Here in Oregon, we have a very creative group that tries to look systematically at the effects of exercise, and it's really quite a powerful effect, I would say. And then there are surgical approaches to Parkinson's disease that you may have heard about, seen videos of. Deep brain stimulation. These are neurostimulators that are implanted into the parts of the brain that are affected by Parkinson's. It turns out that Parkinson's sort of deranges the electrical signals traveling from one part of the brain to the other. And by putting in a stimulator in, you can, this was a happy discovery, but you can normalize that abnormal pattern of neuronal firing and greatly improve the symptoms of Parkinson's disease. There are now, on the research front, there are now non-invasive approaches to neurostimulation. So external stimulators that seem to improve the Parkinson's disease. And then another exciting development, which I think has ways to go, but I think is going to be something to watch for going forward, is a sort of a customizable smart stimulator that can read the state of neuronal firing and adjust automatically. That is where we're headed, I think, is for smart systems that allow the neuronal neurostimulation to be adjusted in response to one's own neuronal firing.
Thank you very much, Dr. Hogarth. I think we are kind of at the end of our time.
Okay.
And I know there's so much more to learn about. I think we've had a real feast today, a real holiday feast. We've had the turkey, we've had the roast beef, we've had lasagna and 3 kinds of pie, a lot more than we can digest. So I think we're going to have to sort of take a rain check on the next session. If you have questions, we're glad to follow up with folks directly. We're so grateful to have members of Congress and their staffers, the ABC membership, and other supporters of neuroscience with us today. And for members of Congress and staffers, we have 2 requests. And the first is to assure robust and sustainable funding increases for biomedical research, and especially neurodegenerative research. Everywhere this is possible. This is the NIH, it's the CDC, it's defense, it's the VA, and so on. And also, for those of you who are on the staff, if your boss isn't a member of the Congressional Neuroscience Caucus, please suggest that they join. A special thank you today to the Congressional Neuroscience Caucus for partnering with all of us, the ABC, the Cure Alzheimer's Fund, the Michael J. Fox Foundation, for hosting this briefing today. There's obviously a lot more to go over. Thanks so much, Dr. Hogarth and Dr. Lidlow. Again, a special thanks to Congressman Blumenauer, and to everyone, warm wishes for a happy and healthy and safe holiday season. Bye-bye.